That's uh, that's good. Okay. Um, we're gonna jump into the neuroscience now. Let me just share back my screen again. And over the next little while, the next few minutes, what I want to do is I want all of this information to make sense um, to everybody. So we're gonna understand what a heat maps are, brain slices, the blood oxygen level dependent signal that's used in fMRI, and what it means to do a subtraction, which is the most common comparison in how you actually typically will view data that arrives from an experiment using fMRI. fMRI stands for functional magnetic resonance imaging. MRIs would be just a static scan, a structural image. The functional means what is actually changing, what mechanisms are happening, what's working, right? The workings of this structure is the functional aspect. Um, so maybe I should actually ask, anybody here have a background in neuroscience and or familiarity with fMRI?
I heard about it, but I don't have any uh, degree in it.
I heard about it. Yeah, okay. And Mary, what about you?
Yeah, I don't profess to say that I've done formal training in it at all, and I've never myself had it done.
Okay. All right. Well, don't worry. You don't need anything. This is designed to bring you up to speed. But I do want to. I think these concepts are not very hard to grasp, uh, as long as you have the opportunity to ask questions to make sure some things I say actually make sense. The first thing that we're going to start with is the bold signal. And so this is bold stands for blood oxygen level dependent. So blood oxygen level dependent signal. So it's a signal which depends on the level of the oxygen in the blood. And what you typically see, this is passage over time, seconds here. This is the activity of neurons. So you get some neural activity, and this is going to be within a defined region of the brain, what would be called a voxel, right, which is the three-dimensional version of a pixel. Neural activity here. Sorry, you can see my mouse moving around. I should double check.
Yeah.
Cool. Okay. So the neural activity here in the red line, bloop, and then you have the bold signal which comes after. And so the first thing you have is first you have a baseline signal. So it's going to be, this is, let's say 0, and then you get a bunch of neural activity. And then on that onset, you're going to increase oxygen consumption, and then you're going to activate it. You're going to increase blood flow. Um, and this is going to lead to a nice big signal like this. Uh, first there's a little dip, and then there's the nice big signal. So that's your signature sort of bold response. This little dip here, and then there's an undershoot afterwards, where you've also used up a lot of oxygen. So that's all the signal is. It's just basically your brain is able to differentially control the volume of blood that passes through different regions. And that's a way for it to cope with the fact that as your neurons are active, your um, your brain needs to power that activity. And you probably heard that the brain uses up a ton of energy, right? Tiny little organ, 2-3% of the body mass, but it uses up like a quarter or a third of all the calories that you eat, which is incredible because you think if you want to lose weight, all you need to do is use your brain a lot. And there's some truth to that. Um, but ultimately, just maintaining your brain is a big deal. And I mean, I have my own theories as to exactly um, why that is, but we don't need to get into that right now. The point is that you can dynamically control how much oxygen and blood and supplies are going to different regions of the brain in a relatively rapid manner. So the time course here is only a few seconds. And so this allows us to then peer into the brain and examine what areas are actually becoming active or less active over time. And if we can do that over time, then we can do it under different circumstances, and then we can compare the different circumstances in order to isolate particular aspects of a circumstance, and then say, okay, the brain is regions A, B, and C are needed for this particular circumstance or activity or function. And the reason why we are able to look at oxygen is because oxygen has a spin and interacts with magnetic fields. And so we're actually able to activate oxygen and let it relax. And then as it relaxes, we can localize its position. And so we can measure where oxygen is inside of the brain. So any questions on this?
All good.
All right.
No, it's awesome. I love it. Thank you.
So the next is the brain slice. Now this should be pretty easy if anybody's ever made a fruit salad or dinner. So if you're cutting up things, you're slicing through them, obviously. But the important thing to mention here is that typically when you look at an image of the brain that you might see in a paper, or even let's say in a popular news article or in a book or something, you have to keep in mind that you're almost never looking at the entire brain. You can sometimes have these surface maps, but it's extremely difficult to visually show on paper or on a screen in a static image what is happening in any three-dimensional structure, because all you have are the 2Ds of the plane to work with. And because of that, the way that we typically present three-dimensional brain imaging data is through slices. And you can cut the brain in different ways. You can cut the brain straight down through the nose, and that's your sagittal cut. You can cut the brain straight through the eyes, right? Level with your, level with the floor, or level with the plane of the earth. And that's your coronal, your axial. And then your coronal is if you cut it ear to ear. That's your coronal slice. And coronal slices are probably the most common one, which are shown. And the key thing to notice here is that depending on where you take a slice, only certain areas of the brain would be visible, only particular subsets. And that's pretty important when you're looking at the brain, because you could see two different images of a brain, and if the slices aren't from the same place, then you can't make really good comparisons. So that's one thing that you want to be primed to check out when you examine papers, is you got to make sure that these slices are actually taken from the same region if you're comparing region to region. So brain slice, that's pretty easy, right? We're good with that?
Yes.
Okay. Now, how to read an MRI scan, the heat map. So this one also, sometimes people have a little issue with this. And so I think the easiest way to demonstrate it is just with a map of the temperature of the oceans. So you look at this here and you can see that you got cold water down by the Antarctic and up in the north. And then as it gets warmer, it moves from blue to kind of greenish into yellow and then red. And this is just showcasing what the temperatures are like inside of the oceans on Earth. And we tend to do this in neuroscience as well when we want to showcase the change in activity of brain regions. And so these are the typical colors that are used, where violet and blue are cold. And then as you get hot, it becomes yellow and then red. And the heat map itself can be chosen from a variety of different colors. So already you see the heat map that I've chosen here with the bold signals on the far left is actually slightly different from the heat map that's shown in the temperatures of the oceans on Earth. So it's that's just something to keep in mind, is that, and the heat map could be reversed, right? Somebody who's just for whatever reason feels like messing around with it could set the heat map to make purple be more activity and more signal, and the other way around. So you always want to check to see what particular heat map is being used and what do those colors actually mean. Understanding that usually fire colors mean more activity and ocean colors mean less activity, usually. All right, is that pretty clear?
Yeah. Yes.
Okay, now this is where we get into something which is definitely not unique to fMRI, but really, really important to understand. And that's the concept of doing a subtraction. So when somebody jumps into an fMRI machine, well, I guess jumping would be the right, the most accurately descriptive word. But when you lie down and you get your head, cemented into this position, and somebody throws a blanket on you to keep you warm because the magnets tend to be cooled, you see how that participant here is staring up at a screen, and there's a mirror there. So that mirror will reflect images onto the screen that's shown above the participant, and this way we can be showing the participant images and we can interact with them. And they have some speakers that they're listening to as well. So they got earphones on and they got a screen in front of their face. And then we can slide them into this giant magnet that will excite oxygen and quantify the concentrations of oxygen in different regions of the brain. And then we make measures. But because the brain is always active, always doing things, right? It's maintaining your memories. It's facilitating your bodily responses. It's interacting with your GI tract. It is, um, you know, contemplating about the future. It is dealing with childhood trauma, right? Your brain is processing and working and doing stuff all the time. So if you were to just image somebody's brain and then show them an image, and then say, okay, here's all the parts of the brain that are interacting or that are active when you show somebody this image, it's not going to be right. What you need to do is you need to understand the context of where that image is shown. You need the comparison as best you can. And usually a good way to do this is to show them first the image that you care about. So in this case, the image that we care about, the thing that we want to understand, how does this affect the brain, is the bird. So the bird is what we are interested in, what brain activities associated with the bird. And so we show the bird to them. But the bird is actually present on top of this striped background. So we can also then show them the striped background on its own, measure what the brain is doing when they see the striped background. And then we can do a subtraction. So we just measure it, the difference. So this is not actually a measurement here that I'm showing. I hope it's, maybe that's not perfectly clear, but this is not a separate measurement. There's only two measurements we're doing, this one and this one. What this is the subtraction of the two. So we'll image the brain and we'll understand, okay, at this point, we get, relative to baseline, we see this decrease in activity here and an increase in activity here. Right, so we're measuring the brain, and then we set everything at baseline, so all the different regions are just relatively consistent. And then boom, we put this on the screen, and then we see an increase in this place and a decrease in this place. And then when we show this image just by itself here, we see there's a decrease in this place. So then if we do a subtraction, if we go this minus this, then the blue disappears and all we're left with is this red increase. And then we can say, okay, it looks like this bird is stimulating activity in this region of the brain.
And what would be the correlation if you would only show the bird?
Well, you can't only show the bird, and that's the point.
But I'm asking about the correlation, like what would be the difference if you would compare the subtraction and then just the bird? Like what would be the difference?
Well, yeah. So this is where my example breaks down, because you can't show the bird by itself. I don't mean to suggest that this stripy image is a thing that we're showing. Um, this stripy image this stripy image represents everything that's associated with showing the bird. So when you show them the bird, there might be, some anticipation. They know the bird's going to come up, and that's part of the what's represented in the stripiness. It could be that the onset of an image, right? So as soon as you show the bird, just the fact that there is something there that was not on the screen before, that that could induce some brain activity or change brain activity, right? So it's impossible just to show the bird. You can't just show the bird, and that's the whole point here.
Mm-hmm.
Um, but what you can do is you can show everything except for the bird, and then do the subtraction, and that's what you're left with here. Yeah.
Yeah, thank you.
Mm-hmm. Okay, so now we're prepared to dive into some fMRI. So there's two studies that I want to um, review, and we're not going to review everything about these studies. Just a couple different findings from each of them. And the first study focuses on the subtraction between focused breathing, which is what Lucas was just doing a moment ago, and mind wandering, which is what we are doing most of the day, and not necessarily a bad thing to be mind wandering. Um, and so here we have this heat map of focused breathing, FB, minus mind wandering, MW, and the regions that mediate the intention that we have, as well as the somatosensory awareness. So if you were just doing focused breathing on a visual cue, for example, then some of the somatosensory awareness regions may not show up. And that's probably one of the reasons why it's I guess beneficial or efficient to be focusing on your breath when you do focused meditations, because in addition to training your brain for focusing and returning to your focus and accepting the fact you've lost focus, you're also able to enhance the ability to sense what's happening in your body. And that's actually really, really important for a lot of things that we won't get into at first, but depending on how much time we have at the end, we can explore later. And so the main regions here that are important for focus are ones that I've circled here in the teal colored, or maybe this is Tiffany blue colored circles, dotted circles. And in particular, I want to highlight the dorsal medial prefrontal cortex. This is part of the brain that is very well known to be involved or activated when you're focusing on something. And it's definitely one of those regions of the brain that seems to be under definitive active control. So it's quite easy for us to activate our dorsal medial prefrontal cortex, which is pretty cool. And as we discover more and more about neuroscience, and we learn more and more of the regions that get activated by different practices, we enhance our ability to have physical control over our minds. And so we can choose what regions of the brain we want to activate and suppress, and having that level of agency over the part of the part of the body that makes us uh, who we are, I think can be extremely transformative and beneficial in a lot of ways. So what you'll see here is you just see this heat map, you see the slices, and then you see these yellow regions. And we know the subtraction is focused minus mind wandering. So it's just the regions that are really involved in the focus that light up here. And then you see how they are. One thing you'll notice is that with this slice here, this is in position 12, and this slice here, it's in position 9. You'll see how the brain is a little bit smaller compared to this one, right? It's the same brain. I mean, this is a composite brain from multiple people, but but you just slice through it at a different position, and so it looks a little bit smaller. Okay. So the second experiment is, we're going to go into a little bit more detail. And this is a really nice experiment, which is examining how the brain responds to emotional stimuli. And I should mention that in both cases, both of these studies are examining people that are novices in meditation. They might have a light meditation practice, but they're not deep, experienced monks or anything. And that's important to mention because it shows just how rapidly and how much control everybody has over their brain if you're just guided with some simple instructions. So in this case, the experiment is going to show the participants a number of different images in very particular order and imaging their brain all the while. But there's two groups of people. One group, they call the mindfulness group, we're given these very specific instructions here. Try to consciously be aware of yourself, of what happens to you at this moment. Do this while expecting the picture and while looking at it. Do not judge. Remain conscious and attentive to your present state. You may focus on thoughts, on emotions, or on bodily sensations. Those are the instructions that they gave one group of participants before they engaged in the study. Now remember, these are not people that are meditating all the time, and in fact, they measured how much mindfulness or how mindful the groups of people are, and ensured that they were balanced according to that. And when they recruited the people for the study, they didn't tell them that there was going to be any experiments examining the impact of mindfulness. So these people are very blind to what's being done here. And then the other group, what they call the basic group, was just given the instructions, expect and perceive the emotional stimuli. That's all they got. So in one case, they're specifically told to expect and perceive emotional stimuli. They're told that they're emotional stimuli. In the other case, they're not told that it's emotional stimuli. They're just told that there's a picture, but they are instructed, they can focus on thoughts or emotions or on bodily sensations. So there's some reference to emotion here. But I personally think this is a flaw in the experiment itself. The fact that they label the stimuli as emotional in one case, and they do not label it as emotional here. I think that is a shortcoming in the design itself. But it's hard to say how much of an impact that would have, and this is not the only study to pull out similar results. So I don't think it is a meaningful confound. All right. Now, the way that they show the stimuli is actually first with a cue. So they prime the participants before they actually demonstrate they show the picture. And they give this little happy face if it's going to be pleasant. So there's a happy face, and then there's a dot, and the dot is used very commonly in these types of experiments just to help people stay focused and to help minimize the heterogeneity in how people may not be perfectly focused on the screen that you're. So you get them, make sure that their eyes are here, and you tell them fixate on this, and it's just way easier for people to fixate on a dot on a screen than just anywhere on a screen that's blank. So happy face, dot, and then butterfly on a flower. That's pleasant, dot. And then sad face, right? Dot, and then unpleasant. I don't know what that is. Is that an ice cream cone that's been dropped? It looks like a lollipop on the floor, or that's like a pile of poo or something. I'm not sure. And then a dot. Neutral is just your flat face, dot, and then cup. And then we have this vertical bar, and this represents unknown. They don't know what's gonna be. It might be a pleasant, could be neutral, could be unpleasant. Don't know. It's gonna be something. And by the way, they don't just randomly pick these images and then decide for themselves whether or not they are pleasant or unpleasant or neutral. They come from a specific database of images that have been compiled and judged by many different individuals to be of certain nature. And then researchers continue to pull images from this database of emotionally tagged images in order to help maintain some consistency in the experiments that are done um, across different laboratories around the world. So they're not just random images. That being said, these images cannot be perfectly labeled, and these images will not necessarily be um, of equal emotional stimulus for different people, right? So if this happens to be an identical replica of the little mug that your grandmother had sitting on her desk when you walked in and you saw that she was had fallen and couldn't get up or something, and you were worried she might be really hurt. Well, that desk, for whatever reason in your mind, might be really negative, right? So it's really critical that they run a lot of images, tons of images, and really take the averages. And then you hope that these things will um, just sort of wash out, just be some noise in the system, but it will not meaningfully impact the results of the study itself. Okay, any questions on the experimental design at all? Anything else I might have missed?
No, all clear for me.
Anything you're curious about who are these people, etc.?
Actually, I was wondering what are the differences between different cultures, because there are different perceptions in different cultures on different pictures and so on. But this must have been taken into account, right? If yeah.
That's a very very good question. Yeah, I mean this study recruited about 40 or 50 people. They would have been local people from the university, probably students, first year students or second year students, um, and it's I don't know where the school is actually, but that's really important. Yeah, the images were compiled by a U.S. group. I think it was Harvard, and I think this study is also done somewhere in the Western world. So yeah, I don't know, but the reality is if there's an image which is consistently showing different brain responses in one culture versus another, that'll show up in these experiments, right? As you'll see in a moment, it's pretty obvious what's an emotion, what's an emotionally charged image and what isn't, just by looking at the brain. Anything else about the design? Okay. Well, first thing I want to show from this study is bringing us back to the dorsal medial prefrontal cortex. So here, you can see they've highlighted it here, and there's some activity. And this is baseline. So this is over time as we go along here. Dot, 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 and then boom, this is the moment where they show the happy face or the vertical bar, etc. So this is the expectation period. There's a certain number of seconds of the expectation period. And remember, we have two groups of participants, those that we call basic and those that we call mindfulness. And they differ only in the instructions that they were provided before being shown all these images. And one thing that you'll notice is that as soon as that dot, remember there's always a dot in the middle of the screen, as soon as that dot shifts to a happy face, sad face, or a horizontal or vertical bar, then there's a jump. There's always this jump right away. And so the expectation of something is already queuing up your prefrontal cortex. Or we're just zooming in specifically on the prefrontal cortex. What we've done is we've drawn what is called an ROI, or a region of interest, around the prefrontal cortex, and just said, okay, all we're going to do is look at the bold signal within these voxels. And there might be 30 or 40 of them or something, 30 or 40 voxels, and that's it. That's what we're examining here in this instance. So what happens in this particular region? And with the dot itself, it's rather stable over the time, but then boom, as soon as that shifts to something, then we get a jump. And then if it's a neutral, right, if it's just a horizontal bar on its own, then we see it just start to drop down. So it's like, boom, it jumps up, process it, okay, it's just going to be neutral. I guess there's no big deal. Drops down, and then boom, it jumps up again at the moment of perception, and then starts to drop down. And that's the case both for the basic group as well as the mindfulness group, when there is a neutral image that shows up. Now, if it's a negative image that shows up, then that's the green bars, right? Negative here. And what you'll notice in the expectation is that in the basic group, the expectation is much lower than it is in the mindfulness group, and how active it becomes. So in the baseline group, here, this is expectation of negative, only this much activity, but the dorsal medial prefrontal cortex becomes much more active when you see that negative, that sign that a negative image is about to show up. And then when it becomes actually the negative image like shown, then it decreases. Then it decreases. From this state here, but in both cases, it just begins to decrease. But there's not really much of a difference in the activity of the dorsal medial prefrontal cortex in the perception of the negative image. The big difference is before the perception. And that difference is also the case when where it's unknown, where there's the vertical line that goes up and down. And so what this is showing us is that in the act of trying not to judge, and because that's what they're doing, if we go back to the instructions that they're given, one thing they're given is says, do not judge. Remain conscious and attentive to your present state. So these people are being primed at this stage here with this unpleasant or with this unknown vertical bar, that something is going to come in and it could be emotionally charged for them. And their prefrontal cortex is becoming more active than it was if they don't get these instructions, potentially in preparation to not then respond very strongly, emotionally speaking, to the images that will follow. Because you don't want to judge it. You're trying to reserve that judgment on what this image means. So that's something interesting where you see here this heightened activity of the prefrontal cortex.
Can I have a question?
Of course.
Uh, as I'm looking at the graph, it seems that if you are uh, not judging, then also the emotions or like your reaction is going down quicker. But it's not the case for the uh, neutral picture, Because in that case, then your reaction is actually going down slower than if you are mindfulness.
Well, I don't know. From the data, the way I see it, it looks like there's no real difference in the basic group and the mindfulness group once after the expectation phase, right? I mean, these blue lines are pretty close together.
Oh yeah.
Green lines are pretty close together. These purple lines are all pretty close together. It is true that this dark purple line here does start to deviate from this one. That could be relevant.
And also the green, also the one for the negative one, it also goes down quicker.
Yeah, here it seems to be going down quicker. Yeah, that's true. Yeah, it's jumping down quicker. I don't know. Um, I mean, let's say if that is true, and there's a couple things. It could be that you just been a lot more, and so you just don't have as much energy to
Mm-hmm.
keep examining it, and so it goes down. I mean, that's one of the simpler explanations, right? Because in both cases, it's the mindfulness group that jumps down
Mm-hmm.
more quickly, right?
Mm-hmm.
But yeah, it's coming. Let's say back down to baseline more rapidly.
And
Let's.
on how many people did was this data collected?
I think there was 40 to 50. I think they recorded 46, and most of them stuck with the study. So yeah.
Mm-hmm. And there is no like, because you're doing like the fMRI of the whole brain, and this is the only part of the brain that is really affected by the meditation. It's like
No, that's not true. I've specifically brought out this one
uh-huh,
to because it ties back to the other study that I showed,
uh-huh.
and it's important for the next bit of data that I'll show.
Okay,
This
okay.
was this is one of the regions that they were interested in, and um, typically when you run an experiment like this, you will look at the entire brain and do that subtraction and say, okay, what regions are having an effect? And then what you'll do is you'll draw that region of interest. So you then select these specific voxels and you'll look back over your data and you'll just examine what happens in those voxels in order to come up with graphs like this. So you just zoom in on this here. Yeah.
Mm-hmm.
Yeah. There's another area of the brain. There's actually 4 areas of the brain that were really affected here. And this is one of them. There's another that's really important that I want to bring up in a second.
Mm-hmm. Thank you.
So that's the area here, the amygdala. Has anybody heard of the amygdala before? It's typically a pretty famous brain region.
Mm-hmm.
What have you heard about it?
It's the actually all the chemicals are actually going out from there, and it's actually affecting how we feel. Is it correct? And also fear and stuff like that. Our reactions, like basic reactions to situations that are happening around us. I mean, like fear and so on.
That's right. Yeah, it's extremely important for the fear response, for strong emotions in general, including positive ones. But it's also able to encode memories of fearful responses um, on its own, and is extremely important for representing the fearful aspects of any memories and things you've had in your past. And it has outputs that can control your responses. And it has direct connections from the visual cortex that allow it to gain access to information even before you're consciously aware of it. So, for example, some images like spiders and snakes will reach your amygdala, and then through your amygdala can cause you to react even before you really are consciously aware of what you're seeing. And so it's a really important structure in the whole fight and flight response, um, how you perceive the world and respond to it. But it's it's a dumb part of the brain because it doesn't have on its own a lot of context. It's not part of the cortex in a way that it can integrate and provide, rational responses, right? Like your response to a snake or a spider or somebody insulting you or something. Your immediate response that might be mediated by the amygdala might make sense from some sort of evolutionary point of view, but in today's day and age, I mean, usually the scream and the jumping on the chair and lashing back at somebody is not the most ideal way to respond, right? So that's the unfortunate thing about it. It's fast, it's dirty, it's fast and furious. That's what it is. But yeah, it's you're right, Lucas. It's very much involved in emotions, particularly negative emotions, and particularly fear. And it's one of the reasons why fear is such a powerful motivator, is the amygdala really has a lot of control. Um, and I think there's very likely a causal relationship between the increased activity of the dorsal medial prefrontal cortex here and the reduction in reactivity of the amygdala here. But as you engage with that, then it becomes easier and easier and easier to do as you practice this. So that circle of thoughts, remember what we're doing here with the circle of thoughts is we're training ourselves to not be upset when we do something we've tried not to do. And doing something you've tried not to do is a negative stimulus in the same way that the images here can be negative stimuli, and your amygdala will react to that. But as you train yourself and you learn to not be upset, you're actually leveraging that activity in the prefrontal cortex in order to activate inhibitory neurons inside of the amygdala so that the amygdala itself does not respond as profoundly to that negative thing that's going to happen, which is losing focus, or in this case, seeing a negative image. And over time, that just becomes easier and easier and easier. And this is what builds up your stress resilience and your emotional control, is actually using your prefrontal cortex to quiet your amygdala in advance of responding to the information. People very often talk about this extra space that they get. There's always a space between when you receive information and when you respond. There's always that space between stimulus and response. And what mindfulness can do is give you a little bit of time, maybe a little bit of extra time in that, before you respond. And not just respond according to the amygdala or other, subcortical nuclei and how they react, but instead quiet that down, leverage information from your cortex, which can give you much more context, an opportunity to empathize with the other person first. Consider that information. Because if the amygdala isn't as active as it is, then it isn't as able to control your response. You're essentially reducing how much influence the amygdala has in controlling the decisions that you make subsequent to that stimulus. And that's pretty cool. I mean, that's profound, right?
I'm just gonna jump in and say that I have no idea how we started this conversation, but I'm absolutely loving it. I was looking at the graph and then I caught like little bits and pieces. I'm not gonna interfere with your flow, but I just wanted to say that I am totally I'm here. I'm totally absorbed into it. So I'm gonna stay for the rest of the workshop.
Great, great. Thank you. You know, the the way to summarize this is done at the cartoon here. So at baseline, or you could say without mindfulness, you get some type of response, and the way that you react is, some activity in the dorsal medial prefrontal cortex and reactivity from the amygdala. And I on purpose use a lightning bolt for the amygdala, obviously, and the circles for the prefrontal cortex. But then with mindfulness, you have a new situation. We have more activity here, and you have less activity here. And this is a much more, let's say, socially suitable way. Not just socially suitable, it's just a preferred state. It's it's preferred socially speaking, but it's preferred functionally speaking, because the dorsal medial prefrontal cortex has connections to other areas of the cortex in ways that the amygdala does not. So your ability to then respond to the world when your brain is set up like this, make decisions, is just so much more improved. And that has incredible downstream effects. Because if you respond, what it's like when you fight with, somebody you love, and they say something, and you react right away, then that gets them more upset, and then it's this snowball, right? Not just with somebody you love, with a stranger in a bar, between countries even.
Mm-hmm.
Somebody want to make a comment?
I'm sorry. I'm humming. I'm humming.
That's great.
Yeah.
So this is I think a good summary, and this is one of the if nothing is really absorbed, if the details are lost in what's being communicated here, the um, the great thing to remember is just that thousands of years ago there was a lot that was understood through trial and error, and now we're getting to the point where we can actually go in and we can figure out precisely what's happening, and that allows us to then fine tune and figure out ways that we can make this stuff work for a variety of different people. And not everybody's going to respond to the same language, the same images, as Lucas was mentioning earlier. But if you can go inside the brain and you can measure, and ideally, if we can find tools to be able to infer what's going on in the brain at scale, then you really have the potential to to help everybody set up their brain in a way like this, which is really so advantageous. But it takes energy, right? It takes calories, takes effort. Mary mentioned, just a couple hours ago about how important energy is in responding to the world, and this is one of the reasons why. Is because without energy, you default down to, let's say, more calorically efficient brain mechanisms, and well, that's just as I mentioned, less preferential, let's say.
If I may have my subjective comment to this, as I'm doing
Please.
like those kind of mindfulness trainings like for a couple of years now, is that it's hard to judge, right? But it's actually changing the a reference framework that I'm using every day, and I can only judge based on how I react to certain situations. But it's also, as I'm getting older and older, it also affects this kind of things. So it's hard to judge what the effect of mindfulness is. But what I can definitely say is that I'm able to focus much more easier, and I am much more present in certain moments. So I'm not like mind wandering into the future or into the past, but really living the moment. And also my reactions to certain situations are less extreme, but it's like even for the positive ones and also for the negative ones. So this is just subjective comment without any evidence, just my personal experience, what it does to myself and how I perceive it.
Is
That's
it is it okay if I'm just thinking out loud because there's a couple of things that are rushing into my dyslexic brain. So you know, the ADHD in me, it's like blah, 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 like all these connections, and I'm like, hold on, just wait, let me catch my breath here. So there's two things that come to mind. One of them is language, and the other one is this in crazy innate ability for us to sense things happening in our body and self-regulate. So I'm curious, you know, because it's been a long time since I've looked at a peer reviewed article since uni. But um, how much do you know about the? I mean, outside of the NLP, I'm just wondering, you know,
Mhm.
we're great with measuring right now, right? We can pinpoint, like we're good with looking at genes, understanding chemical imbalances, hormonal changes, etc. So my head is going, okay, that's great. Is this like this ability to figure out now? It's very logical. It stems from a point of statistical empirical analysis. But I'm trying to somehow, my mind is like, what about before you didn't get, you know, before you didn't have language, first of all, and then you didn't have the tech to play around with. I'm wondering if there's any sort of evidence or research into, you know, when stress comes along, and then when, you know, you feel inspired, then, you know, the idea of quieting down the voice, quieting down the words, and just being able to feel
Mm.
that internal thing. And so I'm, yeah, like my, I'm not, don't even know if there's a question, but there's kind of this weird thought between the flow of how us acquiring language maybe has fucked up, you know, with our own ability to self-regulate just by feeling without having to put any, you know, stats on it. um
That's super
I don't
interesting
know.
question. Yeah, you know, the original origins of language are under, I think, intense dispute right now.
Mm,
So there are people that think it started millions of years ago, and there are people that are like, no, it's like 40,000 years ago, right? So that's really hard to try to match up anything that we know about the brain um, in our past from, you know, let's say we know a little bit about just from the size of the cranium, obviously.
yeah.
Um, but I think, I mean, I think anything that makes us more abstract would intuitively make us less physical. Um, you know, like if you are able to, and especially, you know, in the past, there are a few decades, you know, the last, you know, 5 or 8 decades or something, with so much focus on this, you know, reductionism and people seeing emotion and logic as like two different ways the brain works. I mean, it's just total hogwash, you know.
Yeah.
But yeah, in the absence of language, then I think emotion has a leg up, and logic has, you know, is you know, sort of is tough to get across. So I think you'd probably be right. Language may have done us a disservice in how we feel, um, but that doesn't necessarily mean that it's done us a disservice in, obviously, in how we communicate.
Sure.
Um, and what we gain through communication in terms of our abilities to perform, I think, is far outweighs what we lose in terms of that inherent ability to sense our own bodies. At least that's what I would like to believe.
Mm-hmm. Yeah, it's because I always I think struggling with fast thoughts as a child, like there were sometimes I mean I had a lot of moments when I couldn't get the words out of my mouth, but intuitively I knew exactly what was going on, but I couldn't put it in words. It's an interesting thing. And so a lot of the, you know, a lot of the times, and especially now, I was looking at somatic therapy, like breath somatic release, and seeing some of these people who are just breathing into their body, and then at some point they start laughing hysterically. There's no language to it, right?
Yeah.
And then, you know, and then they start screaming and yelling because there's so much, you know, there's so much load that's kept in their cells, in the intelligence of their cells outside of the mind. You know, it just, it's like naturally I'm like, I'm wondering about, you know, the importance of language, and how it's super powerful. But at the same time, if I'm lying in bed next to you, your body will pick up on my love, right? Or if I'm sitting at a cafe and I'm
Mhm.
talking to you, your body's going to pick up on my energy naturally. Like our body has this incredible ability to speak without words, and to also pick up, you know, like to make sense of things without words. So every single time I absolutely love like neuroscience and psych and stuff like that, because it explained a lot of things. But as I went through my own, you know, journey in whatever hindsight, um, the more, you know, as you said, right, there's you can argue so much, and then you can have people saying, no, language is 40,000 years old, no, it's 3 million years old. So at the end of the day, we have thoughts, which are very hard to validate. We're looking for sort of, you know, some proof, right, in the best way that we know it. And but at the same time, there's this really interesting thing that the body does that, you know, we're kind of slowly starting to scratch the surface. But this amazing neural intelligence that doesn't have a language, it has more of like a feel, like a sensation.
Oh yeah, that's the yeah. I think language is extremely important, but you know, nevertheless is I think its importance has been overstated for many decades.
Mm.
But we're getting past that. You know, that's the great thing about science is how it is able to realize and build on its mistakes, right? Um, but neuroscience is a super young field, right? And most of psychology was, you know, bullshit. So, um, you know, we're still recovering from that, actually, right? I think as a species, from the, you know, the bogus psychology from the 20s all the way through to the 90s.
Mm.
And the whole neuroscience field still needs to grapple with it in many ways. But that you know, that being said, I think, you know, we as you know, even evolution, I think, is the evolution of the brain is really interesting. Like for example, the human brain has gotten smaller over the last few thousand years. I very much think that's because of writing. Right,
Mm.
now that writing is common, people don't need to remember as much. Remembering requires the continual maintenance of engrams, as well as, you know, the physical changes in neuronal structures in order to elicit those engrams at will. If you don't need to remember as much, you don't need as much, you know, just you don't need as many chips, I guess, or whatever they call
Space, CPUs, or yeah.
yeah, you know,
Yeah.
you know, and of course, if your brain regions are closer together, then you can actually physically have faster thoughts. So there's an advantage to in problem solving to have a smaller brain that might be, you know, counterintuitive. Um, but you know, but the evolution of the humans over the past few thousand years demonstrate that. And you might think, well, how is this? Um, Generating, you know, more babies, and evolution actually isn't that simple. We don't simply evolve because of um, the ability to have babies, which I think is a really greatly misunderstood thing in genetics in general. And I'm not just talking about genetic drift, which are things that occur just by chance, etc., and have no effect on reproduction. But there are active processes that allow you to pass on necessary or beneficial um, things in your own life that can enter into your germline and therefore get passed down to your kids. And that's we're just at the very beginning stages of understanding how that might work. And you know, 3 decades ago, that would have been heresy to suggest something like that.
Mm.
So, you know, even things that we think we know really well in science are constantly being rewritten. And one thing I always tell my students is you open up your textbook and 80% of this material is wrong.
Yeah,
And you have
it's
to,
outdated.
no, 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 no. It's even if it's perfectly up to date now, we know that eventually we'll discover it's wrong.
Yeah, yeah.
But but it won't be like flat out wrong. It'll just be missing the mark by a little. So it's like, oh, we thought this dart was on the bullseye, but then if we zoom in closer, we actually see that the bullseye is smaller. You know what I mean? And so the dart was on the bullseye from, looking at, and that's the way that most things are wrong. They're just incomplete, I think. And that, the great thing is to bring this back to the neuroscience of mindfulness and that comparison to the ancient world. I mean, people have been doing this for thousands of years. And it's like they've been doing it because they know it has a benefit, it has some impact. But the great added benefit that we get by understanding these mechanisms is one, helping this become easier to learn and amenable to a greater number of people, or essentially everybody. And two, being able to figure out the nuances and improve on it. But three, being able to decipher once and for all what is actually beneficial and what isn't. Um, and that's really important. And that's also, I think, really going to help acupuncture too, because they do a lot of brain studies with acupuncture, and they're able to understand, okay, when I stimulate this region, when I put a pin into this region of the body, it activates this area of the brain, which is neighboring this region of the brain, and then that, you know what I mean? And I think that if the West is able to accept acupuncture in a more intimate way, there'll be just a lot better understanding and resolution, and then you can get a lot more precise and continue to update the maps, right? The acupuncture maps. Anyway, um, so that's exciting stuff. I don't know how much time we have left. I don't need to rush or anything at all, but I do want to bring this back to the circle here. So we learned about two important regions: the dorsal medial prefrontal cortex, which we know is very much involved in focus. If we just do a simple subtraction of focus versus mind wandering, it's one of the four main regions that truly lights up reproducibly. And I bring it to the attention here because it's not something that just showed up in one or two studies. This is a well reproduced finding. When you focus, you recruit the dorsal medial prefrontal cortex. And then as you lose focus, you get this reaction from the amygdala that says, damn, I lost focus. And that's natural. But you're gonna notice that you lost focus. Once you do that, reactivating the dorsal medial prefrontal cortex. And then you're going to use that as a way to then quiet the amygdala to say it's okay. And probably these are long range glutamatergic connections. You're releasing a neurotransmitter which tends to activate other neurons, and you're activating neurons in the amygdala that actually serve to quiet the amygdala down. Right? Most brain nuclei will have their own internal inhibitory system that basically is able to govern how active they are. And so we stimulate that inhibitory network, it quiets the amygdala down, then you start to focus again. And you'll notice as you do this meditation that each time you lose focus, if you're truly taking the time and the effort to not be upset with yourself when you lose focus, every single time you lose focus, it bothers you a little bit less, a little bit less, a little bit less, a little bit less. And that is training to get control over how you respond to negative stimuli, regardless of whether or not it comes from your own brain, it comes from your own body, or it comes from the external world, external in quotation marks. So, and those are the three main sources of information for your brain, right? Internal, the brain itself, from the body, and from outside the body. But your brain can't really distinguish them, right? It's just electrical signals from the brain's point of view. So, any questions on this? Something this makes sense?
Why cannot our brain actually distinguish between the different sources of information, like between our inner body, outer body, and inner?
Well, your brain can, because we know it, right? We can determine. So if we know there's a difference, if we can even describe there's three, then there are. So the way I said it is not precise. What I should say is the chemical nature of the information is identical.
Okay, yeah. So if you're actually doing the fMRI, then it looks exactly the same, and you cannot distinguish whether it's like where it's coming from.
that's precisely right. And equally importantly, that means that the training that the brain receives is the same. So the just by doing this circle of thoughts in your own head, you are preparing yourself to respond in a more mindful or less reactionary way, even to negative stimuli that will come from inside or outside your own body. That's what I mean by having that chemical uh, similarity between all three sources of information.
And I think it also plays like there's a very popular kind of like concept going around saying that, you know, like your brain doesn't understand positive and negative. Like if you think negative thoughts, it's the same sort of thing happening there. And I think I remember like the first time someone told me this, I was totally blown away. It was in a workshop many years ago, and they said, whatever it is that you do right now, do not think of a blue elephant. And then everyone thought of a blue elephant, right? Because it's like the brain doesn't understand do not. It's exactly what you're saying. The firing is there, like, you know, the language is the meaning that we give to that stuff.
But I saw that the chemical reaction is actually different if it's positive or if it's negative. It's the same, Dr. Besh?
Well, I mean, there's different reactions, and you may even, you know, depends what you mean by positive and negative. In I think the point that was being made here is that um, you know, you can't prime the brain between do not and do. That was the idea, right?
And I think it's also like when the language gives, you know, because it's like if your language is, you know, negative, then your body starts producing x amount of, like, even though the circuit is the same, the path is the same, once you start attributing that language, you know, that internal dialogue towards whatever it is that is happening, then your body starts to react to it. And then maybe that's why you're alluding to, Lucas, like your body will respond differently depending on how you interpret this, you know, like universal neural link. Am I like saying this right, or am I totally going on?
Well, I mean, you know, there's definitely going to be a different signature in the brain if you show people negative or positive images.
Mm.
You know, you can tell if somebody's looking at a pleasant thing or an unpleasant thing based upon what's going on in their brain. So there's, you know, just regional differences. Um, you know, but I think the if you look at mood, um, you know, the good and bad mood is probably just more or less of different transmitters, right? So it's not like there's one system for negative, one system for, you know, positive, or let's say pleasant and unpleasant would probably be the better words to use, because positive and negative could mean so many different things, right? Could mean has not, could mean yada yada yada. But pleasant versus unpleasant is a little more precise. And those are that's typically the words that are used for to describe valence, right? Which is how good or bad you feel.
Mm-hmm.
And that's, yeah. So, but ultimately, it's let's just say it's complicated, and it's also idiosyncratic, right? Like it's wrong for us to um, pretend like the brain works the same in every single individual, right? And by necessity, these experiments are done with multiple people, and then you have the averages of them, right? But even things as basic as, you know, what side of the brain are you processing language on? That's not the same in all people, right? So there's really no that's one of the great myths, I think, of neuroscience, is that there's this left right brain sort of dichotomy, and it's just it's garbage. I mean, it's not true at all.
Mm-hmm.
You know, there's no person on the planet that uses one side of the brain more than the other, right? And even the regions of the brain that we consider to be left and right are all messed up all over different people, right? So who knows what's going on in your head? Um, and because it's all different in all different people, then, you know, on average, somebody's brain is definitely going to have something different. And anyway, yada 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 yada.
Cool. Okay, I'm good. I'm done with the questions or suggestions or observations.
um, I think we're past time already. There's
Yeah.
but I'm done actually. This is done. I have this little moon circling here to like help maintain another meditation. But just a quick review, I'll just say, number one, stress resilience can be improved with mindfulness training. All right, that is a fact. All right, at this point in history, we can say from the body of research being done. Two, we learned about some important things of fMRI: heat maps, slices, bold signal, and subtractions. And my fantasy, people will walk away and actually remember one or more of these important elements about fMRI. And then thirdly, um, strengthening the prefrontal cortex can help control the reactivity of the amygdala to negative stimuli. And that's a neural basis for why mindfulness can help us build stress resilience and emotional control. But more broadly speaking, the punchline is, we can train the brain in extremely precise ways. This is just one example for how focused meditation, and in particular the circle of thoughts, is able to help train the brain uh, and liberate, let's say, a more mindful experience, but let's say a more nuanced experience, and of life. But this is just one of many examples, right? So we could have dove into other experiments that show other things, and that's truly an exciting. It's really an exciting time to be alive from that perspective, because we're only going to discover more and more and more, and that's going to reap a lot of benefit. That's my email, and of course that can be found on LinkedIn as well. Well, thanks everybody.
Awesome.
So,
Okay. All right. I think yeah, we're pretty much at the end of the day. We had the consistent number coming and going and moving around and flowing. So that was interesting. I had technical difficulties. I'm coming in from my phone right now, but I'm just I'm gonna come back and watch this from the beginning in our replay, and I'm very excited to bring everyone together. And uh, some of you have actually said that you want like a proper social with the speakers, because everyone is super interested in what everyone else is doing, and they want to, it's like they want to be friends. So we will probably like circle back to that at some point. But this has been really great. I mean, thank you for being here and sharing all of the wisdom and energy and time that you have with us. And namaste. That's all I can say.
Great, thanks, Andrina.
Thank you.